This is a production of Cornell University.
Yeah, I want to talk about the genetics and breeding of resistance to Fusarium Ear Rot in maize. And the title is not, I mean, it's kind of a joke. It's unfortunately, it's not fully a joke because, I'm looking back. I've been doing this for, I think we started these experiments 16, 17 years ago. And I just feel like if you've been working on a topic that long, you should know a lot more than I do. You should have made a lot more progress than I have. But so this is largely a history of various attempts we've made at trying to understand this disease resistance, mostly ending in failure. But hope springs eternal, and the sort of the last half of the seminar is the stuff we're doing now, which we think might be reasonably effective. Figure out how to advance my slides. There we go. So this is a description of the interaction that occurs between this fungus Fusarium verticillioides, which is the causal agent of Fusarium ear rot, and maize. So if we start over here, hope you can see my little pointer, but this fungus likes to overwinter in crop residue, and when seeds are germinating, it can infect germinating seeds. And one aspect of there is a disease called Fusarium seedling blight, same fungus. I guess it's a different disease because it attacks these little seedlings as they're coming out of the ground, and it can kill them. But what happens actually much more commonly is that the fungus will colonize this emerging seedling and actually become a systemic symbiont almost. I like to think of it as basically like an endophyte. It grows in here, but it can colonize into the developing ears, and it can take over the developing kernels and give you the ear rot. Now, that's one way this can happen. It's probably not the most important way. Presumably, the most important way that Fusarium causes the ear rot is through the silk channels. So you get some wind, rain splash, the conidia get onto the silks and travel down the silk channel, and they come through the styler canal. This is where the silk attaches to the developing kernel, and that's presumably the mechanism for how they get in there. And then I'll show you these pictures of the starbursting in a second, which is the result of that. Same fungus also can get into the stalks, and it could have been sort of systemically there, or you could have some insect feeding that helps it gets into the stalks. It'll rot up the stalks and cause lodging. Also, you can have insect damage on the ears. That wounding from the insects allows the fungus to get in and colonize that ear. So just to start things off with, there's a lot of different ways this fungus can get into these kernels and cause this disease or infect other parts of the plant. So again, this is just a little bit better picture of this of the Fusarium ear rot symptoms. This is the classic one. This is starbursting. And this is the one you'll probably see most commonly. You have these little streaks that are actually caused by directly by the fungus itself growing under the pericarp. If things really get out of hand, that fungus can really just destroy the entire ear, and then all kinds of other necrotrophs can get in there and cause a lot of damage. Reduces yield and grain quality. Here's what the stalk rot looks like, and of course that contributes to lodging, so that's a problem. Here's the Fusarium seedling blight. This is a paper where they took seeds this is from a maize diversity panel, and they infected or they inoculated the seeds with Fusarium verticillioides as they were germinating. And they also had a control where they didn't inoculate. The controls basically look like this one scale here, but then you have these sort of increasing scales of damage to the point of where, basically nothing even germinates because the fungus colonizes that seed and kills it right away. So same fungus, same host, and of course we've screened this same panel for the Fusarium ear rot, this thing that you see in the mature ears. And so an obvious question came up is there a relationship between these two aspects of the disease, or these two different diseases caused by the same fungus on the same host? So I ran a little correlation. On the x-axis, we have the seedling blight score. On the y-axis, we have the ear rot score from the field. And I'm pretty sure this is as close as I've ever been in my career after measuring lots and lots of correlations. This estimate is as close to 0 as I've ever seen. So, I mean, there's really no relationship between these two things. So right off the bat, you can see that even though it's the same fungus, the same host, something is very different about this kind of disease from it versus the adult plant. We also did some studies where we tried to look at the correlation between the adult plant ear rot and stalk rot. And we kind of started that experiment, and after splitting a few hundred corn stalks and measuring a preliminary correlation, it was not high enough for me to want to continue splitting corn stalks because that's just way too much work. So we kind of gave up on that. So it does look like these kind of components, this ear rot, the stalk rot, and even the seedling blight are really kind of different diseases, even though the fungus is the same. So, that's the first thing I still don't really understand. Well, what is going on with this fungus? It's kind of crazy. It's in this very diverse genus of fungi, some of which are pathogens of corn and other cereal crops. And we tend to get this Fusarium ear rot caused by Fusarium verticilioides, in the southeast and in the southern parts of the country and in the tropics. It's very common. In cooler areas, the Gibberella ear rot caused by Fusarium graminearum seems to predominate. So they seem to be, these are very similar fungi, very similar diseases, but it looks like the pathogens have different sort of favorite environments. Then there's other species in the genus that are endophytes of grasses that are essentially symbionts. They don't seem to be pathogens at all. And even in corn. So maize can be infected, not show any symptoms, or as I shown you, Fusarium can kind of go wild and cause ear rot. So even in symptomless ears and kernels, you can find this fungus. It's often there just hanging out. And, some folks in the mycology, pathology community consider that there's a continuum between endophytic lifestyle and pathogenic lifestyle. And Fusarium is somewhere in the middle of this continuum. And it can sort of sometimes be more like just an endophyte and other times be like a pathogen. And I think that's why this is such a complicated disease. And we don't really understand what tips the balance to make, in a particular case, to make this fungus become pathogenic. So focusing a little more just directly on the ear rot, so I showed you these starburst symptoms. The other thing that happens with the Fusarium verticilioides growing in the kernels is it produces this secondary metabolite, Fumonisin, which is a mycotoxin, and that's been linked to cancers and liver and kidney diseases, both in humans and in animals. So that's a bad thing. We want to make this disease kind of go away because of the direct damage it does, but also because of this indirect effect of getting the grain contaminated with these mycotoxins. And here's our big problem. We still have, in my opinion, near zero understanding of the biology of resistance. There's no immunity, there's no major effect R genes, and I'll show you, but there is quantitative variation. But we don't really understand the biology of that. So, as I said, the most important route of infection of the fungus into the developing kernels is through the silks. So fungus gets on the silks and it can move down the silk channel and it can get in through these where the silk, this is the silk attachment scar. There's a little opening there early in development where the fungus can get in. And this, in this paper, Duncan and Howard developed a Fusarium verticilioides expressing GFP so that they could image where in the kernel this fungus was going after it infected. So you can see there's a lot of fungus right there at the silk scar, and then it makes these streaks. It grows down the streaks, and it's under the pericarp. It's between the albumin and pericarp layers. And if you look later in development where you see the star bursting, this is not actually star bursting because this is before you can even see the star bursting, but the star bursting occurs exactly where this fungus is growing because the fungus is getting in there and actually destroying some of the tissue in that region. So this is a beautiful paper where they did very detailed biology of this, and they tried to compare more resistant, more susceptible varieties for, what's different between them, but it was actually quite difficult to tell. One difference was in the silk scar, but it's pretty clear when you try to drill down to these very detailed levels that each of these little components is probably only playing a small part. You have all these environmental factors, and there's just tremendous complexity in what this fungus is doing. So the fungus is kind of has this strange lifestyle, and then it produces this mycotoxin, like a number of other these serial infecting pathogens. Probably worldwide, fumonisin is occurring at relevant levels more frequently in grain samples than probably any other mycotoxin. This is a paper where they looked at grain samples from different parts of the world and whether they were above some threshold that's specific to the mycotoxin, depending on how bad the mycotoxin is. So there's aflatoxin. Aflatoxin is really bad in parts per billions, so it's really, really bad. But fumonisin is bad. You have to be in parts per millions, but it occurs so much more frequently that it might, in fact, be a bigger health hazard than aflatoxin, just because it's so widespread. And here's North America, South Africa, 92% of the samples. So it's very, very common. So why is it making this thing? The obvious initial idea was this is probably a virulence factor, but this is studies from other labs. It appears not to be a virulence factor for Fusarium Ear Rot. It may, in fact, be a virulence factor for that seedling blight. There is evidence for that, for it to infect the seedlings, that fumonisin helps it do that. But it doesn't seem to have any effect on the ability to colonize kernels and ears. And then there's this other issue of you get more mycotoxin in some environments than in others. So this is a sample of commercial hybrids grown throughout the U.S. corn growing regions, and no artificial inoculation. This is just commercial hybrids under regular production conditions, and they got the grain samples from them, assayed them for fumonisin, and you can see there's this general trend for an increase in the levels of fumonisin as you go to the south and toward the east. So the hot spot in this study was here in North Carolina in our corn growing region near the east coast. The other thing I'll just point out, sort of an aside, is that even the lowest levels in this study, well, there was two sites that were below the FDA recommended limit for food in most feed, but all the other sites were above it. And again, that's without artificial inoculation. This is presumably a very common situation. This study was done in the early 90s, so it's pre-BT, and it's clear that having a BT gene will reduce the levels of fumonisin because it reduces the amount of insect damage on the ears, which reduces the amount of disease and fungus in there. But it doesn't solve the problem. We still have, particularly in North Carolina, we still have too much fumonisin in our corn. So there's all these things going on. This is a little slice of a review from Charles Bacon that, again, points out that there's this sort of, Fusarium verticillioides has this sort of dual lifestyle as both a pathogen and a symptomless endophyte, which really indicates the complexity of this relationship. And again, something can alter the balance of all these factors that are affecting this relationship that sometimes push the fungus into producing getting more aggressive, causing more ear rot damage, but also producing the mycotoxin. But again, since we don't understand exactly what's causing this, it's hard to really design sort of informed biological approaches to resistance. So let's talk about resistance and what are the genetics of resistance. And just the common theme here will be that it's highly polygenic, lots of environmental influence. The resistance is pretty variable across populations, really importantly, there is no known immunity. So we evaluated the entire USDA PI collection of inbreds, and there was not one in there that we couldn't at least get a little bit of Fusarium ear rot on. So there's nothing like an R gene for resistance, but there are these quantitative differences in how much ear rot will be observed in different lines. Our big problem here is we can phenotype this thing, and the phenotypes are informative, but it's very labor intensive. It's a lot of work to get good data on this. So there is some kind of resistance, but what is it? What are the genes involved? These are the sorts of questions that we've been very interested in. And this brings up this issue of it's clearly not primarily conditioned by like R gene mechanisms. So the question is there even any active response that's part of the resistance? Or is the resistance just sort of a constitutive level? It's like as long as the plant has these factors, it's not going to get infected. Or is it both? Is there some component of both of these, that there's constitutive factors, and on top of that, maybe there is some active resistance? But again, I don't think we know the answer to that question. Been a lot of number of QTL studies, and I'll point out Laura Morales' study in Rebecca's lab that was published last year. That was done in several NAM populations. We did this, long back in the dark ages. I mean, the bottom line is you can map QTL. They vary among the populations. It's like the classic story. And there's no really major resistance genes. And even by major, I mean, if you want something that conditions 20-25% of the phenotypic variants, we're typically not finding something like that. So the original idea, let's map some QTLs and we'll do marker-assisted selection, which is sort of how I started. That kind of went by the wayside. That didn't really work. So then, right, GWAS came along. Let's try GWAS. Maybe we'll do a better job. We'll look at more diversity. And again, we went to the point where we tried to look at everything in the PI collection and do a pretty large-scale GWAS on that. And we have a paper where we've identified 7 SNPs associated with the ear rot resistance. They all had pretty small effects. The vast majority of the genetic variation is just picked up by that polygenic background term. So, and then there's some more studies that have been more recently. People will do another GWAS and then they'll find something different, which also has small effects. So again, it's not like we've hit on anything that you would want to turn into a marker to use for selection studies. There have been studies on differential expression in response to infection. So far, these have been limited to 2 host genotypes. So, I think we can't really generalize too much from those studies as to what's really going on. So here's the challenge. We're trying to breed for resistance, and I've just admitted to you that I don't really understand anything about what the resistance is. I don't understand the biology. I don't understand what's going on with the fungus. So, it's from the point of view of understanding the biology, I'd say really I've made very little progress. But we have this sort of, I'm sort of a half-time plant breeder, so I can always just switch hats and say, well, I'm not going to worry about being a geneticist and understand all this. I'm going to be a breeder, and I'm just going to select for resistance. And it works. So as long as we have reasonable data, which means, replication, multiple environments, and we don't need a huge number of environments. We've typically gotten away with 2 reps, 3 to 4 locations, 1 year, 2 years data. That's pretty good. We'll get heritabilities that are above 50% on the line means. So we think we can make some progress. We have evidence that we've made progress. And this, again, tells you that the genetics are really important, even if we can't identify a single gene that's, really interesting to talk about, or we don't really understand what the pathways are, but we know there is genetic variation that underlies this resistance. And then a couple other little tidbits that are sort of sideshows, which is kind of goes back to this idea where measuring, inoculating all these things, measuring everything, which we do visually, but it takes a long time. It's just a lot of work. So if we could find some shortcuts, that would be great. One of the shortcuts we found for Fumonisin contamination is that there's a very high, strong genetic correlation between the ear rot and Fumonisin contamination. And we've validated this over numerous studies. So selection against ear rot, which is still difficult to measure, but it is cheaper at least than measuring Fumonisin. We should get a pretty good indirect selection response just by selecting against ear rot. And then Laura and Rebecca, they identified this relationship between test weight and higher test weight, kind of more dense seed, is correlated with reduced ear rot and Fumonisin. And test weight's, it's pretty easy to measure. It's pretty fast, and it's also not just visual. You put it in a machine, and the machine gives you a number. Still, we're limited by the labor-intensive nature of phenotyping, but sometimes these shortcuts are pretty useful. So here's sort of like the workflow of doing this, evaluating these things in the fields. We have to we have to culture the fungus ourselves. We worked with Gary Payne in the plant pathology department when we started, but Gary has retired, so we're kind of like abandoned orphan plant breeders with no plant pathologists to help us anymore. That scares me because, again, I don't really know what I'm doing. As far as I can tell, I know how to culture Fusarium, and so far that's what we're doing. And so we culture our own Fusarium, grow it up. We make these liquid inoculum solutions, and then we'll go. So we actually start over here. We'll, about a week after the ears silk, we'll go in and inject them with this solution with the fungal spores down the silk channel, which we think mimics a lot of what goes on in the real world. A week later, just to be sure, we'll come back and we'll actually wound them by poking them through the husks and injecting the solution between the husk and the ear that way too. So we get a little bit of wounding. So we're kind of trying to mimic kind of both of the major pathways that we think are occurring to infection. Then we have to harvest all this stuff. We bring them into the seed lab, lay everything out. All these ears get scored visually, 0 to 100% symptoms. And then if you're doing just ear rot, we're done. Then you have your data, you're good. But if you want to get Fumonisin data, you have to run everything through the sheller. This is our seed lab setup for this kind of operation. It's pretty, for us, this is big scale. It's a lot of work. You have to then grind all these samples up, and then you have to run ELISA assays, which are a couple of bucks per sample, so it's not super cheap. And also this, we've enriched these grain samples for this toxin that will, destroy your liver and give you cancer. So everybody has to work with PPE, and it's really a pain. So a little bit on these, indirect trait correlations that we think are at least favorable. Number one here is that we've shown this in a number of studies now, that if you evaluate inbreds, inoculate the inbreds, evaluate the ear rot or the Fumonisin in the inbreds, you can predict the top cross hybrid performance of those for resistance pretty well if your tester is a susceptible tester. And then we went back and we looked at this test weight, and we thought, well, what would be really easy is if you just evaluated test weight of the inbred line, and could that have any prediction of a hybrid of that line for the resistance? And actually it worked. You have to inoculate to get the best correlation. If you inoculate the lines, that's on the x-axis here. Here's the test weight of the inoculated S1 lines. And on the y-axis, we have the FER is Fusarium Ear Rot of their top cross hybrid. And on here, we have the Fumonisin levels of their top cross hybrid when they are inoculated. And you can see the correlations are pretty decent. So that's all great. So, we thank Laura and Rebecca for pointing this out to us, because, going forward, this is probably something that will allow us to screen larger numbers where we just don't have the people power to evaluate all these ears by eye. It does appear, though, that we need to inoculate to get reliable results. So my student Eric Butoto is almost done with this paper, which we'll submit. soon. But this, going back to this, how are we going to do a better job of breeding without really understanding anything? We have all these great genomic tools available to us now. So can we use genomic tools in the absence of really any biological understanding? And I'm not saying that's the situation I want to have. I'm just saying that is the situation we're in. And that's where genomic selection is should be a really good method, because the trait's highly polygenic. Genomic selection is good for that. It's really a pain to phenotype. So if we could get marker data, that should be cheaper and easier than that. So for all these reasons, we're thinking, now's the time. Let's see if we can pull off an actual genomic selection study on this thing. So this is Cornell. So I'm talking to a Cornell crowd. So you guys know what somebody who really knows a lot about genomic selection looks like. There's Jean-Luc. And then this is how I look, So I'm a little bit old school. But this is this situation, even my old school breeder self thought, okay, now's the time. I mean, this is the situation under which genomic selection should be really useful. And I'm just kind of old and tired of having to inoculate all these things and evaluate all these ears. So maybe genomic selection is going to be a good approach. So here's how I've turned the corner and latched myself to the bandwagon just as it's leaving the station. Back up one little step to tell you the population where we're implementing this genomic selection is a population we developed. We call it refuse for resistance to Fusarium. And it's basically, we crossed a set of lines that have good adaptation to our region to a set of lines that have good resistance, mixed the whole thing up, random mated. Everything is tropical or non-stiff stock, so that any lines that come out of this, we can cross them to a stiff stock tester as a potential hybrid. And we already ran through 3 cycles of S1 phenotypic selection. That was done some time ago. And this is the evaluation. This is phenotypic selection evaluation. In this panel, we have ear rot, and the blue is the cycle 3. The sort of red-pink is cycle 0, where we started. So the ear rot has gone down, and that's by a pretty significant amount by these 3 cycles of phenotypic selection. This is fumonisin. And again, the fumonisin content in the grain gone down. That's due to selection, so that's great. Here's yield, and yield didn't really change, that's fine. We didn't want to ruin yield. We weren't really selecting for yield. We just didn't want to ruin it. So phenotypic selection works. We're happy. We can measure it, but it's a lot of work. And the other key thing here is that each of these cycles of S1 selection required 2 years. There is a lag between, when we have the S1 lines, we're evaluating them in the field. We harvest all those ears. We bring them back to our seed lab. For example, the experiment we've done this summer, we haven't even started scoring ear rot. We've already prepared all our seeds for winter nursery. We don't have any data yet. So we lose a generation. We can't use that next winter generation. And so what we have to do is next summer, we'll have the data, we'll do the intermatings, and then need another generation after that to get the S1. So we end up using 2 years per cycle. And that, that's a lot of time. So then we started thinking about, okay, how can we go forward now with genomic selection? Let's build a training model using the data we have in hand. So this is my student, Tiago Marino, who's now a breeder in Brazil. He evaluated this using 250 lines from that cycle 3 population that we had data on multiple locations across 2 years. And then as a validation set, we just generated 200 new random lines just by selfing out of that same population. So new lines, and we phenotyped them in a new year. So new lines in a new years, but it's the same generation, the same population that they came from. Everything was genotyped. Talk about that in a second. And then, then we just did basically it's our cross validation where this is our training set and this is our validation set. The genotyping, just as a little aside, We used a modified GBS. This is developed in Randy Weiser's lab. Randy was a student of Rebecca's, and he helped us with that. What it does is it's a further reduction of complexity, so we don't score as many SNPs, but we have much less missing data. We can call the heterozygotes in these early generations better. And what should happen is, because you're doing size selection as part of this, we should get a more repeatable set of SNPs every time we redo this in a new cycle. In that the base population now is we're calling cycle 3. There's very little population, there's a little bit of some family structure here, but there's actually very little population structure. We measured LD. There seemed to be enough LD to tag resistance even with just 6,000 markers. Here's our result. So again, predicting into a new year, new genotypes. I don't want to explain all this. This is some technical, boring details. We had 2 different partitionings of the data. And you can see it's kind of okay. It's not great. It's not terrible. For Fumonisin, we do a little bit better on Fumonisin. The heritabilities tend to be better on Fumonisin because it is measured by machine. The correlation between the predicted and observed is 58%, and that's an accuracy of 64%, so it's okay. Ear rot's not as good. I don't think we're quite as accurate there. But even with a correlation of 0.37, we started thinking, man, if we could turn 2 cycles a year of genomic selection, as opposed to 1 cycle every 2 years with phenotypic selection. In other words, we could run 4 cycles. We could run 4 cycles of genomic selection in the time we can do 1 cycle of phenotypic. That ought to give us some advantage to genomic selection, even though the accuracies are not super great. So again, if you can get direct phenotypic observations from well-replicated good trials, that's the best thing to select on. But that takes too long. So if we can do genomic prediction faster, that should work. And over time, we should get a greater response to selection. And this is, again, I'm talking to Cornell, Mark Sorrell, Jean-Luc. They've been pointing this out for over a decade. So we're just following in your footsteps. Here's what we've done. We took that base population. We selected the best 20 lines, like we normally did, purely on phenotypic data. So we then just bifurcated the program. We have the PS is the phenotypic selection thing, and we took the S1 remnant seed of those, intermate them, randomly self, so you get a bunch of S1 lines out of that. We evaluated that 2 years ago in 3 locations, inoculated, did our whole shebang, and identified the 20 lines with the lowest Fusarium Ear Rot. We then, those got intermated and generated a new cycle we call cycle 5 of S1 lines from that. On the genomic selection side, we had remnant S1 seed. Well, every time I have a little star, we did genotyping and selection. So in this generation, we selected among, you're segregating S1s, we can genotype those and get some boost from genomic selection. We've intermated these guys, made a cycle 4 S0, intermated these guys, made a cycle 5 S0, and then we actually shifted to alternating selfing and intermating. I'll talk about that in a second, why we did that. Well, the other thing is in that sort of evaluation of the potential study, we used the data from the first 2 years to predict the other 200 lines from the last year. But for this thing, we actually took all the data and use that as our training set. Here's the gory details. This is for the genomic selection experts there. This is like the gap between theory and the practical actually doing of it, at least in our hands, which are not great. First off, if we could get our breeding value predictions before flowering, we could have made twice as much genetic gain as we actually do. This is like a purely technical point. That's like a major bottleneck. Industry knows how to do this. They genotype seeds, and they actually make selections before they even put things in the field. We're still planting seeds, taking leaf samples, extracting the DNA, making libraries, sequencing, and you got to run it through the bioinformatics, and then you got to get the predictions. Some of that is easy, some of that is hard, unless you have you're an expert with a lab that's really good at this stuff. And there are labs that are. My lab's not. So, this was challenging for us. So typically we were getting the breeding value predictions back, around harvest or even after harvest, but we'd get them before the next cycle, but not before flowering. So in those first cycles, what we did was we just randomly intermated pairs of plants and we genotyped them. And then at harvest, we kept track of the male and the female. We had the predictions for the male and the female parents. So we could predict the breeding value of that cross. The problem was we would have individuals that we had used in the intermatings that we tried to genotype, and then the genotyping failed for some reason. So they dropped out. So we just, it's not a big deal for this. We just give them a 0 for our breeding value because we don't know what it is. And then we just moved on. But that was a little bit sad that, on top of like missing the chance to select before flowering, we have this second problem where this isn't really genomic selection as it should be done. This is genomic selection as it gets done by people who don't really know what they're doing, like me. Having this data though, for like quantitative genetics nerds, the data are a lot of fun. And it is fun to mess with these data. So this allowed us to implement optimal contribution methods, which are basically a way you can predict how much inbreeding you're going to get in the next generation. And we just put a restriction that we weren't going to increase the inbreeding coefficient more than 5% per generation. That might have been too loose. Probably was. You'll see. And then the hard part about implementing that optimal contribution is, that has to be calculated on sets of genotypes, not on individual genotypes. So then we implemented a genetic algorithm. Again, we didn't invent any of these things. It's just like we're pulling all these tools that other smart people have invented and trying to put them to use. But that was fun. But also we have parents that are contributing to our next generation where not only do we not know the breeding value, we don't know the relationships. So we can't guess how much inbreeding they're contributing. So that actually really threw a wrench. We had to assume that they weren't related to anything. That's clearly a bad assumption. So we were not controlling inbreeding very well. So those later cycles, we shifted to alternating and selfing and crossing because actually the genetic gain is predicted to be the same from that in this condition. But what it allowed us to do is by selfing, genotyping, and then only crossing selfed progenies that we had the genotypes on it, we didn't have to worry about all this missing breeding value information. Oh, this is a really sad story. I don't want to dwell too much, but at some point we actually failed on our modified GBS, and then we were in a hurry. We had to get data, so we had to pay LGC a lot of money to genotype stuff. And the LGC genotyping was good, but it's expensive. So, clearly for someone like me who's not a genotyping expert, like either I need to, outsource this to another lab, pay a lot of money, find another lab that's really good at genotyping and partner with them. But I think DIY, in my case, is a bad idea because I think where my failings are really on the genotyping side. Since we do have the genotype data on the selection cycles, we can look at what's happened in terms of the relationships and diversity. The red dots here, that's the base population, and these other colors are the later generations as we're pushing selection. And actually, we saw this right away in the first generation. Basically in one generation it wasn't complete separation, but one generation of genomic selection really pushed the allele frequencies very hard, very fast. That was a little scary, and it made me a little nervous at first, but we just kept going. You'll see this is the last cycle in blue, and it's kind of like trifurcated into like subgroups. I think that's because we were not going to allow it to just keep going this way in the dimensions because that would be too much inbreeding. And you can see it on the kinship. matrix. Here's the cycle 3. Here are the later cycles. And then this is our last cycle here. You can really see we've got some family structure popping up. So we didn't do a great job, even though we tried, we didn't do a great job on managing inbreeding. This is my student Eric Butoto, who's working on this. So we have samples now of this initial population and both the genomic and phenotypic selection end populations, and we have samples also of the intermediate cycles. We planted all those this summer. Everything was inoculated, everything's harvested. It's all sitting in the seed lab right now. We don't have the data yet. So sorry, I don't really have the answer, but we're still pretty optimistic that the genomic selection was at least reasonably effective. Whether it was better than the phenotypic or not, I mean, we got to get the numbers. I can't really just eyeball these things and tell. We'll genotype the phenotypic selection samples just so we can see what happened to diversity in those. And then we tried to measure a bunch of, ear morphology things that we thought might be related. We'll also look at test weight, just as a way to see which of these other sort of indirectly correlated traits might have changed with selection. Just the last little bit of theory, going back to theory, this is a paper from Melkinger's group a few years ago, where they show that if you start with a training set here at cycle 0 and you start implementing genomic selection without retraining, so this is just continuing to use the base population training set, your cumulative gain, you're still doing pretty good up through 4 or 5 cycles, but it really starts plateauing after that. So clearly, no matter what we decide to do going forward, we're going to have to retrain the model. But I think before we decide where we go next, I really want to get the data, answer these questions on how well we did in terms of genetic gain from phenotypic genomic selection. How much diversity did we lose? Is that, how big of a problem is that now? Should we shift away from recurrent selection to something like biparentals and pedigree selection? Everything's kind of on hold until we analyze these results, and then we'll try to make an informed decision going forward. So just thanks to all the folks who've done all the work. Jason's my technician. Eric and Tiago were the grad students who did the most recent work on this, and then David, Charlie, Megan, and Leilani. This is like a chain of grad students. Leilani came in 2003 and learned how to do all this stuff, and then she taught Megan, and Megan taught Charlie, and Charlie taught David, etc. So it's been nice to have this chain of grad students who know what they're doing, since I don't really know what I'm doing. But I'll stop there, and I will take any questions.
Great. Thank you so much, Jim. The floor is open. Feel free to use the chat or just open your mic and...
Ace laughed at my joke, so I appreciate that.
Here, Rebecca, this is Ed. Do you have any guess as to what the biology is on the test weight one? I mean, is it protein packing on the starch granules, or removal of water, or something, making it more hypoxic or something?
I don't have a hypothesis. Rebecca might. I don't have a hypothesis. I will say, on top of whatever is actually sort of intrinsic to the seed of that line, that correlation is definitely better when you inoculate and measure the test weight. So some of the decrease in test weight on more susceptible lines, I think, is due to the fungus getting in there, damaging the kernel, and it must be that the whatever mass the fungus is less than the corn, that it's like destroying the kernel before it can fill properly. So part of it is actually the direct result of infection, but Rebecca may have other ideas.
Well, I'm just wondering if the test weight would reflect both the genetic differences in density or test weight, and then also the fungus induced ones. And we kind of cottoned on to that hypothesis when we were cracking open a lot of corn kernels, and there's pretty much a hole in the kernel. We did those early CAT scans, and we could say, like, oh look, it's got a hole in it. Basically, this is more for aflatoxin, but well, we see we can really clean up grain by sorting out the lightest fraction. So I'm wondering how does that affect you if it's kind of you've got a confounding effects of inherent density of the different genotypes and the induced density difference.
Yeah, I don't know. I mean, one thing you could do is you could measure test weight with and without inoculation and make some kind of index of loss of test weight. But right. I actually think it is a bit of both, where I think if you have a line that tends to have higher test weight, those probably do tend to be more resistant, because we see the correlation, it holds even without inoculation. Although even without inoculation, you do get some fungus.
No, I've done that little causal diagram too. Yeah, I think it is that the denser stuff is more resistant. It's trying to colonize a diamond, basically. If you've got a real flinty and sperm.
So we showed this result. Actually, Tiago showed this result at his exit seminar. And for the folks who don't know, Major Goodman is the corn breeder here, and he's really old school, and he's funny. So his question was, since you want higher test weight, he says, why don't we just grow popcorn everywhere? So this sort of idea, you can take things to logical extremes, and it can get absurd, but clearly
that was kind of the thrust of my question too. But we had this also, like, by sorting it, sort of stratify the sample and look at the ratio of the weights of the lightest and heaviest. The toxicity of the two. You can get deeper into which one you're looking at, or inoculated, non-inoculated. Although non-inoculated, North Carolina doesn't count for much because it seems to be no such thing. I do have to say, you've got a bunch of questions coming in.
Okay.
Shitaya, you want to go ahead and ask?
Sure. My question is about when the fungus lives as an asymptomatic endophyte with the plants, does it still produce secondary metabolites? Thank you.
My understanding is that yes, it does. There are cases, like, you can get. This is the scary part of it. You can get a lot of corn seed that has no symptoms of the ear rot, or you can't tell that there's fungus in there. There is fungus in there, but you can't tell visually. And in those cases, you can still sometimes get reasonably high levels of the mycotoxins. So yeah, it's not a simple one-on, one-off thing, although the mycotoxin levels do tend to get higher when you see more ear rot. That is also true, but it's not a perfect correlation.
Jean-Luc, want to go ahead and sing out your question?
Well, yeah, Jim, sorry. you showed this population structure in that last generation. There's nothing in particular about optimum contributions that I can think of that would generate that, but can you explain a bit more?
I'm going to share the screen so I can show the slide you're talking about. This business in particular, you can really, this is the last cycle. I don't know that I have a great explanation for it. I think, the other question is, I'm trying to think now that I've given this seminar, you should ask me what was my population size, and now I'm actually blanking on that. We tried to get 384 per cycle, but there were enough dropouts that it was below that always. So we were probably in the 250 to 300 range of individuals.
And there's number of selection candidates.
Yes, and then we were recycling crosses between 20 parents. So if we were picking F1s, we would pick 10 F1s from 20 parents, or if we were picking S1s to go to the next generation, it would be 20, and then we would. So I thought that would have been enough to avoid the extreme inbreeding. Well, it's not really extreme, but to avoid the inbreeding that we're seeing. But I think probably something happened in the previous cycle where We just didn't account for it, and probably on some of these samples that weren't genotyped. So it's not the fault of optimum contribution, it's the fault of not having data on everything that's actually contributing to the next generation. So we were probably putting in something like half SIB, possibly even full SIB type sets were contributing to that. And then when you made crosses between, you know, this guy that was a full SIB of that guy, they crossed to related things. You started seeing more of a family structure. I don't think that should happen. I think it's sort of a coincidence, but I mean, I was not expecting that. When I saw that, I was like, what the heck is going on?
Okay, thanks.
Over to you, Ace.
Yeah, so thanks, Jim. It's great talking to you again and hearing your thoughts on this because, you know, we also struggle with these issues all the time. So I guess like we know that, you know, that Fusarium pathogens can exist symbiotically and like non-pathogenically. And so I think kind of that question about like how they transition into actual full-blown like infection as opposed to just like existing in symbiosis seems to be like when plants are more stressed out, they just come down with like a lot of intense infection and a lot of toxin. And do you feel like because, you know, a lot of actual like resistance genes for Fusarium that maybe like breeding goals could be better set towards like overall ways to reduce plant stress as opposed to actually breeding for mycotoxins per se?
Yeah, I mean, yes, I think that's all true. I mean, the best thing you can do, in my observations, is don't work with inbreds. I mean, as soon as you make a cross and you get a reasonably vigorous hybrid, disease goes way down, and it's just this general. The plants are more robust. They're more tolerant of stress. So some of the things we've been doing is sort of intentionally stressing the plants. That's why we actually want to select on inbreds, because we get so much more variation. Yeah, I think selecting for stress resistance would be really helpful. I don't know that that's easier. I think it's actually probably harder than what I'm doing. And so, you know, that's where the challenge comes in, to really do, you know, to really get good, precise measurements of traits that are truly related to stress resistance. I think that's hard. I think that's actually harder than what we're doing.
Yeah, I mean, but you can always like look at things like NDVI and other easy and see how well those kind of things are correlating with mycotoxins.
Yeah. Right. So if it turns out easy, I mean, this is the thing where like if somebody said, "Oh, I could march a drone through your field and give you ear rot data," I'd love it. It'd be you know, that'd be the next bandwagon. I would be the last guy to jump on. You know, I guess I'm a little skeptical that NDVI. I don't know. It's empirical. We should try it. See what happens. It's worth trying. I'm just not, you know, like all these things. I want to see data before I'll really jump on it.
Yeah, I have this idea because Danilo in our lab is looking if NDVI is a better predictor for like yield depression due to like actual, you know, like foliar. I mean, he's working with foliar pathogens than actual like scoring pathogens themselves.
I think on foliar pathogens, you're gonna have really good correlations. Whether that's gonna translate so well into the ears, I don't know. It's a problem. Yeah, it's a problem having a disease that's hidden inside the husks, so we can't image the thing from the sky. So it's recalcitrant to that whole thing. And as far as I know, until maybe somebody will invent some wavelength that will go through the husks. That'd be great.
Yeah, but because of the connection between the stress resistance and the vulnerability or the susceptibility to these colonizers, I mean, that's what makes me feel a little better about measuring them in near isogenic lines too, that you're not confounding just adaptation and maturity and susceptibility.
And Ace has seen our fields, our experimental fields. Like, you know, the variation in NDVI is, you know, genetics is probably a small proportion of that. A lot of it is just we have really not very uniform fields at a lot of our stations. And it's just the nature of the bees. So a lot of this work is getting driven by places with good uniform soils. A lot of the Midwestern states, I think, can do this much better because they have better uniform soils.
So, Jim, just coming back to that issue of endophyty, I'm not sure I've ever seen really compelling evidence for what you and others sort of always refer to of the switch to toxigenicity under stress. Because it seems like all the cases in which you have toxin being produced are when you have kind of an introduction of the fungus into the styler canal, and as you described it, or into the roots of the seedling. But once it's an endophyte, this shift to a toxigenic state, particularly in the ear, like, do you have reason to believe that occurs? I mean, I feel like the colonization's enhanced by stress, but I'm not sure about the switch to toxigenicity.
Yeah, I don't know. I mean, the quote I showed was from Charles Bacon, and I think that's where he's saying that it may be, a weakened plant is more susceptible to the toxin production. I guess I don't know.
Yeah, I haven't seen that, because that would imply like a change in the amount of toxin per unit fungus, where it seems like it's really just a change in them. Anyway, I haven't spotted any evidence to say there's sort of this disconnect between fungus and toxin. Stress it and then suddenly it starts producing toxin, or starts becoming an ear pathogen. I haven't really seen that. Anyway, anybody else want to raise any issues here? Anyway, if not, sort of, we're just on time. So Jim, I'm just really very grateful to you, and I was expecting a lovely talk and some
Sorry you didn't get it.
Got it all. So thank you so much. Really appreciate it.
Yeah, we've got one last question from Arcadio in the chat.
Oh, there we go. Arcadio, over to you.
Sure, sorry. I was just wondering if, are you looking at all that RNA expression data to see if there's different kinds of genes being activated that might be missing in some of the parents that could explain the basically inability of GWAS to identify things, because just some parents not having a given background to allow a positive response to the disease?
Yeah, so we are thinking about this now. We haven't done it. I have a student who's submitting a NSF, whatever, pre-doc proposal, and this is almost exactly what he's proposing, which is to take a reasonably big sample of the typical maize diversity panel and look at expression response to inoculation in that panel, and of course the beauty of that panel is that Ed's group has already generated just fantastic tissue specific transcriptome data from a lot of different tissues. So that it's a nice way where we could say, basically how much information do we gain in terms of being able to predict whether you're going to get more resistance or less resistance from this active response versus just, taking the data that Ed's group has already generated, whether it's in seedlings or in an adult plant, but not challenged with the fungus. That may, the different, so really focusing on the differences in expression among genotypes that may have, maybe just constitutive. How much of does that predict the resistance versus how much gain do we really get from looking at the kind of active response that you could measure? That's something that it's probably a hard experiment to do well, but it's something that we're thinking about. It might be useful to do.
Thank you.
So there's like 4 minutes until the graduate student chat starts. So if anybody wants to take a quick stretch or break of any kind, now might be the time. But meanwhile, let's a huge thanks to Jim, and enjoy that chat with the grad students. And thanks so much, Jim. Okay. Really appreciate it.
This has been a production of Cornell University, on the web at cornell.edu.